The cytoskeleton of the cell is formed from protein protein fibers or protein structures that support the cell, hold the organelles in place, enable the cell to change shape. They just have different things that they do. But there's three types of protein fibers, that form the cytoskeleton. And they are microtubules, which are the thickest, microfilaments, which are the smallest, and then intermediate filaments, which are medium size. The microtubules form cilia and flagella. Cilia and flagella are composed of microtubules. And they also form what I call the spindle fibers, or the spindle microtubules, that attach to our chromosomes in cell division and pull them apart so that each new cell that's formed gets the same number of chromosomes as the parent cell. So microtubules function in cell division, and they also form cilia and flagella. Microfilaments are the smallest of the protein fibers, so they're small. And they support the cytoplasm and help determine the cell's shape. Some are involved with movement. There are certain microfilaments in muscle cells that enable muscle cells to contract. So muscle contraction is caused by microfilaments called actin and myosin. And then intermediate filaments provide support to the cell. One type of intermediate is keratin, which of course provides protection to skin cells. So intermediate filaments are like medium diameter. They're in the middle between microfilaments and microtubules. And this just shows you a picture so you can see and compare between the different ones. So the microfilaments are shown here. You see how small they are as compared to the microtubules, and then the intermediate filaments. So microtubules are the thickest, the microfilaments are the thinnest, and the intermediate filaments are in the middle. Centrioles are structures found within the centrosome. So the centrosome is where the microtubule formation occurs during cell division. Those microtubules, I call them spindle fibers or spindle microtubules. The microtubules that are a part of cell division are formed in the centrosome. The centrosome contains two centrioles. They normally are lined up perpendicular to each other, and they are composed of microtubules. And so the centrioles and the centrosome are involved in cell division. Microtubules also form cilia. Cilia are little hairs that line the surface of certain cells. Some examples I can think of are cilia line the cells that line our airways, and they can move mucus and trapped materials, trapped particles in the mucus. They can sweep that mucus up and out of our airways. So there's also cilia that line the tubes where sperm is formed. Before the sperm becomes active and can move on its own or swim on its own, the cilia in those tubes can actually move it. But then once the sperm becomes active, sperm cells have a flagellum. Each sperm cell has a flagellum, and it's just used for swimming. So flagellum is one long strand, where cilia is many little shorter hairs. Both are made from microtubules, and microvilli are folds within the plasma membrane. And what they do is increase the surface area of cells that contain them. So there are kidney cells, there are intestinal cells that function in absorption, and they're going to be lined with microvilli so that they can absorb even more. So a little bit about DNA. All of our DNA is divided up into genes. So the DNA that we have is found in our 23 pairs of chromosomes, and each chromosome contains a certain amount of genes. Well, a gene is a piece of DNA. It's a sequence of nucleotides, because remember, DNA is composed of billions of nucleotides. So a gene is a sequence of nucleotides, and those nucleotides provide instructions for making a protein. So that process is called gene expression. When a gene codes for a protein and makes a protein, it's called gene expression. The first part is called transcription. In transcription, this happens in the nucleus of our cells. DNA is copied into a single-stranded molecule called messenger RNA. The messenger RNA leaves the nucleus, enters the cytoplasm, and attaches to a ribosome. And this is where translation occurs. Translation is when messenger RNA is used to produce a protein, because the messenger RNA contains the message for the specific amino acids that make up that protein. So a DNA molecule consists of nucleotides. Nucleotides are the monomers of DNA. The DNA is double-stranded. Each nucleotide has a 5-carbon sugar called deoxyribose. It has a phosphate group, and it has a nitrogenous base, which is either A, C, T, or G. And you can see this is an overview of gene expression. The DNA in the nucleus, a piece of that DNA called a gene, is used to make a messenger RNA strand, which leaves the nucleus and attaches to a ribosome. And then the messenger RNA is used to code for the specific amino acids in the protein. Transcription is the first part of gene expression, and it takes place in the nucleus of the cell. And it is when messenger RNA is formed. It's when DNA is used to form messenger RNA. And then during transcription, all this tells you is that during transcription, if the DNA nucleotide is an A, then it's going to pair with a U. And so what we're seeing here is one of the DNA strands is being used as a template to produce this messenger RNA. So if you have an A in the DNA, it's going to be a U in the messenger RNA, because RNA contains U instead of T. So A is going to pair with U, G is going to pair with C, and C is going to pair with G. But if the DNA has a T, then messenger RNA is going to be an A, because so A always pairs with T and C always pairs with G. But in RNA, if the DNA nucleotide is an A, it's going to complementary pair or match up with a U in RNA, because RNA does not have a T. All right, so A is going to complementary pair with a U. A is going to pair with U, G is going to pair with C, G is going to pair with C. T will pair with A will pair with U, G will pair with C. That's how it works. So A's pair with T's unless it's RNA, then A's pair with U's. And then C's pair with G's always. So the code, basically, in messenger RNA, every 3 nucleotides is called a codon, and it will code for a particular amino acid. So a codon is 3 nucleotide bases on the messenger RNA that codes for an amino acid. And then there is a molecule called transfer RNA that has an anticodon. which is complementary to the codon on the messenger RNA. So that transfer RNA matches or pairs up with the messenger RNA, specifically at the place where that amino acid belongs, because a transfer RNA transports amino acids to the ribosome and the messenger RNA. So I was looking to see if there was a picture, but I guess not. Okay, so basically the transfer RNA knows where to transport the, here we go. Here's a transfer RNA. So basically this is how it works. The transfer RNA has an amino acid on it. This particular transfer RNA contains the amino acid aspartic acid, and it knows to put the aspartic acid right here in this spot because it also has an anticodon that is complementary to the messenger RNA codon. C pairs with G, U pairs with A pairs with U. So the codon is GAU. The codon on the messenger RNA is GAU. GAU is the codon for aspartic acid, which is the amino acid that comes next in the protein, in the polypeptide chain that's going to form our protein. So the transfer RNA, this orange transfer RNA, is going to have the anticodon that pairs with the codon for aspartic acid. And then it's going to transport the aspartic acid right here to where it goes on in its special place in the protein. Okay. The cell cycle includes basically a dividing phase and a non-dividing phase. So the cell cycle is going to include interphase, which is the non-dividing phase, and then it's going to include the dividing phase called mitosis. Interphase, when the cell is not dividing, is going to consist of the G1 phase, the S phase, and the G2 phase. In the G1 phase, all of the organelles in the parent cell are copied so that the daughter cells, the new cells that are formed after cell division, they each get a copy of the same organelles that were in the parent. So G1 is when the parent cell copies its organelles. S is when the DNA is replicated, so you have two copies of the DNA in the parent cell. So when it divides, each daughter cell gets a copy of the same DNA that was found in the parent cell. Then you have the G2 phase, and that's when the cell prepares to divide by producing those spindle fibers or microtubules that are going to help separate the chromosomes when the cell divides. So here's our cell cycle. We have interphase, and that starts with G1, the first gap phase. This is when routine metabolism or chemical reactions occur in the cell that normally occur. And then you also have the organelles. All the organelles in the parent cell are going to double. Then we have the S phase, which is when DNA replicates. S stands for synthesis. And the G2 phase is when the spindle fibers or spindle microtubules are produced that are going to play a role in cell division. Then you go into the M phase or mitosis, and that's divided into prophase, metaphase, anaphase, and telophase. Mitosis is division of the nucleus of the cell. Now, each human cell contains 23 pairs of chromosomes. In other words, a total of 46 or 23 pairs in every human cell except for sperm and egg. Sperm and egg are called sex cells, and they're only going to contain 23 total chromosomes. Now, 22 of our chromosome pairs are autosomes, and the last pair, the 23rd pair, are the sex chromosomes. So the 23rd pair of chromosomes, the sex chromosomes, they determine whether a person is genetically male or genetically female. They're genetically female if they have two X sex chromosomes, and they're genetically male if they have an X and a Y. So if chromosome pair number 23 is two X's, that's going to be a female, and if it's an X and a Y, it's going to be a male. So mitosis is the division of a parent cell and the formation of two daughter cells that are genetically identical to the parent. So it's like cloning, okay? Mitosis is divided into 4 phases: prophase, metaphase, anaphase, and telophase. And so we're going to look at pictures now. This is interphase, which shows that the DNA is still spread out in a form that we call chromatin during interphase. And during prophase, the chromatin condenses into chromosomes. So during prophase, this is what happens. Chromatin condenses into chromosomes. So we can actually see the 23 pairs of chromosomes under the microscope. We also see microtubules that we call spindle fibers attach to the centromere of each chromosome. The centromere is where they're connected. Centrioles will begin to move to opposite ends of the cell, and the nucleus will disappear. That includes the nuclear membrane and includes the nucleolus. All of that will disappear. So this is what prophase looks like. The nuclear envelope is disappearing, or nuclear membrane is disappearing. Chromatin has now condensed and formed chromosomes. Centrioles are moving to opposite ends of the cell, and then these fibers that you see extending between the centrioles, and then the ones that you see radiating out away from the centrioles, those are the microtubule or spindle fibers. They're going to attach to the centromere, which is the place where the two chromosome strands are attached. So this chromosome has 2 strands called sister chromatids. Each one of the strands is called a chromatid, and they're identical. They formed during the S phase of interphase. When the DNA replicated, it was basically single-stranded chromosomes that replicated and formed double-stranded chromosomes, which are now attached at a centromere. So these spindle fibers, these spindle microtubules, are going to attach to the centromere of every chromosome. And then during metaphase, the chromosomes will line up in the middle of the center of the cell at the metaphase plate. And it is the spindle fibers that will kind of move them toward the center of the cell and line them up. So now you can see in metaphase the spindle fibers attached to the chromosomes at their centromeres. And all of those chromosomes are lined up in the middle of the cell. Now the sister chromatids are going to separate. This is anaphase. So the sister chromatids will separate during anaphase, and they're going to separate because the microtubules or spindle fibers will shorten. And that's what it looks like here. When those fibers shorten, they pull the sister chromatids to opposite ends of the cell. And then during telophase, you basically can take telophase and everything that happens in prophase, you can reverse it. So if you can learn what happens during prophase, telophase is the reverse of that. So now the chromosomes that condense during prophase are going to unravel, and they're going to become more diffuse and spread out. They're going to become chromatin once again. During telophase, you're also going to have nuclear envelopes or nuclear membranes forming around the daughter cells. And the spindle fibers are going to disappear. I don't see where it says all of this, but basically you end up during telophase, toward the end of telophase, you have the cell splits, and that's called cytokinesis. And so you're going to have 2 daughter cells produced at the end of telophase and cytokinesis. And each daughter cell is going to have 23 pairs of chromosomes. There'll be single-stranded pairs, but there's still going to be 23 pairs, or a total of 46. They're just going to be single-stranded and not double-stranded. Okay, so this is what telophase looks like. You see the nuclear membrane or nuclear envelope reforming. The spindle fibers are gone now. And then by the end of telophase and cytokinesis, the chromosomes are now diffused and uncondensed back into chromatin. And you can see the nucleus again and the nucleolus. And so that is the end of the cell cycle. This is a picture of some human cells that are in the various stages of the cell cycle. This is what interphase looks like. Prophase, you're going to start seeing the little purple spaghetti strands, is what I call them, but you can actually see the chromosomes. They have condensed. And then you can see in metaphase, they've lined up in the middle. In anaphase, they're pulling apart toward opposite ends of the cell. In telophase, I see telophase, I always look at telophase under the microscope. When the parent cell begins to indent in the middle, then it's telophase because cytokinesis has begun. But in anaphase, the cell might be longer. It might not be as rounded. It might have formed more a longer oval shaped, but it hasn't started indenting, hasn't started splitting apart yet. So anaphase is here. That's when the sister chromatids separate. Telophase is also when cytokinesis begins. And then you can see cytokinesis and telophase have gone a little further, and eventually the two cells will split in half and the process will be over. Now, there is a section on cancer, so I'm going to stop here with this recording.